Sorry for the interruption, guys. So we're talking about the neuromuscular junction here. And the main events that happen here, and if you learn this well at this point, they will facilitate your understanding what happens in the nervous system between chemical synapses that are established between two nerve cells. All right, so this is a chemical synapse, and whenever a signal arises from a nerve cell to the axon terminal, which is going to be an action potential, that will cause the release of the content of vesicles that are present at the axon terminal there. And so these neurotransmitters contained in vesicle is going to be released in the synaptic cleft, which is this space really between the two types of cells. Two types of cells are not in contact. There is a space in between, okay, but it's relatively small. And so this neurotransmitter, which is the acetylcholine, travels and binds to specific receptors. These are sodium channels, actually sodium-potassium channels, very different, very unique. They have nothing to do with any other type of sodium-potassium channels that we talked about. These are ligand responsive, and they respond to acetylcholine. And in response to the binding to acetylcholine will lead an influx of sodium, a massive influx of sodium inside the muscular fiber. All right, so what happens afterwards? Okay, so first of all, I want you to focus on the three important factors that play a role here. There are these vesicles, okay, that contain the neurotransmitter. There are the channels on the muscle fiber, and there are the ions that play a role here, especially the sodium ions. Okay, these three things you cannot not know for me. These are main factors that play a role in the neuromuscular junction. And so, as I told you, when an action potential propagates, there is a release of acetylcholine, which in turn will determine basically an action potential that propagates through the sarcolemma. Remember, the sarcolemma is the plasma membrane, and remember of the muscle fiber, and remember that it extends in these T-tubules. Okay, so there's a lot of sarcolemma around that surrounds all the myofibrils. And ultimately, remember, what you want is release of calcium. And calcium here is contained normally inside the sarcoplasmic reticulum, which is all surrounding this blue thing here, surrounding all the myofibrils. Okay, so let's look at this in more detail. We said that upon action potential that arises at an axon terminal, there is exocytosis that happens. All the vesicles that contain acetylcholine will release that neurotransmitter in the synaptic cleft. And that neurotransmitter binds to specific unique channels, sodium channels, that are present they're very localized. They're not present throughout the whole sarcolemma. They're only present right there in this plate. And remember the shape of this side. At this side, the sarcolemma is kind of like represented these invaginations. Okay, elsewhere it's flat. And so right there are these channels that, upon binding to the neurotransmitter acetylcholine, will determine, will let a lot of sodium come in. Sodium comes in because it is more concentrated in the synaptic cleft versus the inside, so there is a diffusion basically that happens there. And therefore that determines that influx of sodium determines the inversion of the resting membrane potential. So remember, normally, I'm gonna draw it again. Okay, this is the plate where we're talking about the you have normally a negative net charge inside and a positive net charge outside. This situation, upon binding of the acetylcholine neurotransmitter, is inverted, okay, to where now you will have a positive here and a negative outside. All right, and so that is a depolarization, right? So there is a depolarization, but it's very local because there isn't enough sodium still coming in, okay? Let's say, and these channels will close pretty soon. And but that local depolarization that has happened here propagates in the vicinity, and right in the vicinity of a muscular neurojunction, there are in the plasma membrane of the muscle fiber other types of channel. Please pay attention. These are two different types of channel. You should remember, if you have studied, we looked at these voltage dependent sodium channels, voltage gated sodium channels. They have a door here, right? They're different, and they're different from these ones because they are voltage dependent, whereas these ones were ligand gated, okay? And this, upon depolarization, the local depolarization will activate this channel because they're voltage gated, they're open, and now a massive sodium influx is going to happen, and a massive depolarization. You see how it's all positive. This should be normally all negative charge, right? So the depolarization now can spread out real fast, and it spreads through the T tubules, which run parallel to the cistern of the sarcolemma, right? And the sarcolemma here contains calcium. Now, the whole point is that you want to release that calcium because you want that calcium to act on the myofibers, which are over here. And so the depolarization that runs through the T tubules is the signal that determines the opening of these calcium channels, which are also voltage gated, okay? And so now you have the release of calcium. That's the ultimate goal, right? So it's a very fascinating system to finally regulate muscle contraction. Here is a very nice video. Watch it again, please. If you haven't watched it, watch it. Watch the videos. They're very, very well made. Okay. And remember, calcium will displace this troponin. Will bind to troponin and will displace tropomyosin. Now, therefore, the mechanism of contraction can happen. And we have already described this, okay, the growth bridging. I'm not going to go over it again. All right, so again, it's not that complicated if you keep in mind the main channels and molecules that take place in this action. Remember that acetylcholine needs to be removed from the cleft after it's been released. This removal happens pretty quickly, and happens especially enzymatically. There is a specific enzyme that will chop the molecule up so that, you know, another action potential can stimulate, therefore, again, the muscle fiber. Otherwise, there will be the stimulation will be too prolonged. All right, again, watch the videos, okay? And there are several more videos, watch the videos, okay? In the last part here, we talked about the various types of muscle fibers. Skeletal muscle fibers are not all identical. There are at least 3 types that we can recognize. Some contract slowly, some contract very quickly, and some of them undergo fatigue um, very quickly. Um, these are the ones, the fast glycolytic fibers type 2B, the ones that contract very quickly. These give us the sprint. If we have to take a short run but very intense, are the ones that are going to act but and be activated, but they're very easily fatigued. Okay, and at the histological level, they also stain differently. Okay, so these are the three types that we're talking about. All right, here's a table just to summarize. Don't worry too much about all these details. Just kind of like if you can keep this in mind for me. All right, so then we talked about the muscle twitch. A muscle twitch is something artificially tested in lab where you take a muscle and you give a stimulus in the lab and you can appreciate a contraction. Okay, so it's a single contraction. It's not something that happens really in real life. And when you do that, you appreciate though how the muscle fiber, that muscle can actually contract. And when we do that, we can observe this contraction pattern when there is a latent period, basically between the time that you stimulate, you give this stimulus, and the actual contraction, there is a latent period, okay? And where nothing happens. Then there is your contraction time, and then there is your relaxation time. Okay, this is described as muscle twitch. You can see how as the action potential spreads through the sarcolemma, the muscle twitch then follows. Okay, and so there is a period of contraction where calcium is released. You know that here is a calcium that plays a role. The relaxation time is a period of time where muscle contraction slowly goes down because calcium is uptaken back into the sarcoplasmic reticulum. And if you look at different types of muscles, they present different type of muscle twitch in terms of time. Some contract quickly and relax very quickly, and some contract more slowly and relax more slowly. So not all muscles present the same curve concerning that muscle twitch. And we also talked about the fact that muscle contraction can be regulated basically by two factors: the frequency of stimulation and the intensity of stimulation. We also talked about types of muscle contraction. Okay, don't worry too much about this, but frequency and intensity of stimulation can regulate muscle contraction. And here is an example of how you can regulate muscle contraction by frequency of stimulation. The most common situation is where if you stimulate a muscle frequently over and over, you have this situation where basically your contractions, your tension over time goes this way. Basically what is showing this curve is showing that your muscle fiber is not relaxing completely before the next contraction. It doesn't go down here. See, it contracts over and over, okay, and reach a point where the contractions are more regular. It gets more intense and then over time, but then at some point it becomes regular. This is known as unfused tetanus. This is the most common type of muscle contraction. There is a situation where basically you can create a so-called fused tetanus. This is a situation where you have stimulated pretty much the muscle fibers so much at its maximum to where basically the muscle is contracted, and it does not have the opportunity to relax, and so it keeps being contracted in a contracted state. Okay, and so you are here on a maximum tension of your muscle. Regarding the intensity of stimulation, you can play on that, and of course the intensity of stimulation playing on the intensity of stimulation, you can recruit less or more muscle fibers, and you can reach again a maximal contraction by, of course, recruiting all the muscle fibers from pretty much from this point on. Okay, you have recruited all the fibers that are in that muscle. All right, so I think this was the last one, the last of this lecture, just on the first lecture. Now I'm going to go over the second lecture of the muscular system, and I'm going to go a little bit quicker here. So we talked about cardiac muscle tissue. You must be able to recognize this histologically, okay? And be able to describe the characteristics of this type of muscle tissue. This is involuntary, okay? Very different from the skeletal. It is intrinsically controlled by a pacemaker, a set of cells that set the pace of the contraction. This, just like the skeletal, is striated, and the cells here are branched. One very important characteristic, typically mononucleated, but sometimes dinucleated, as shown here. And so remember this histology could be presented to you. Now, the one important difference here between skeletal and cardiac muscle tissue is that the cardiac muscle cells, they communicate through specific types of, if you can call them synapses. These are electrical synapses, basically are gap junctions which connect directly one cell to the next. And if you look at it histologically, you will recognize where a cell starts and where it ends, okay? Because there are these lines here, they stain pretty strongly. These are known as intercalated discs, okay? And these intercalated discs are the levels at which there are the gap junctions, which are channels that span from one cell to another. Through this channel, you can have passage of ions very quickly, okay? And so it turns out that all the cardiac cells are in tight communication, fast communication, because of these channels, and therefore they form a so-called electrical syncytium, which means a unit. Okay, so it's an electrical unit there. It makes a lot of sense because all the cells in the cardiac muscle tissue, they have to contract simultaneously. Imagine if portions of the heart and muscle tissue, the cardiac muscle tissue, would not do so. They would say one region would contract at one time, and another region at another time. There could be big trouble for the organism. And there are several pacemakers actually in the heart, but the one that really overtakes and has the fastest and strongest signal and action potential is sinoatrial node, which is located over here, okay? And this receives innervation from the brain, and now you should know which region of the brain. Remember when we talked about the medulla oblongata last time that regulates heartbeat? See how things here connect, okay? And one other thing I wanted you to appreciate about cardiac muscle contraction versus skeletal muscle contraction is, first of all, the duration of an action potential is prolonged in a cardiac muscle. And this results in a very prolonged contraction. And that's important because you don't want a very fast contraction, relaxation, because the function of the heart is to pump blood. And so basically, if you didn't have that time, that extended relaxation time, you wouldn't have the opportunity for the blood to be pumped everywhere in the body. Okay, so very different type of contraction. And what elicits the contraction of cardiac muscle is the same exact thing that elicits the contraction of skeletal muscle, is calcium, and this is always the case also for smooth muscle. So you cannot go wrong there. You don't have to remember different things. It's always calcium. Now, the mechanism by which calcium is released is different. And I really talked about the mechanism of calcium release in cardiac muscle tissue. You don't need to know that, okay? But we're going to talk about the mechanism of calcium release in smooth muscle tissue, okay, which also is the mechanism to elicit contraction. Now, you have to know where smooth muscle tissue is found, of course. And, of course, here we're talking about involuntary activation. The one striking thing here, when you look at smooth muscle tissue histologically, is that you don't appreciate striations. That doesn't mean that myofibrils aren't present. It just means they're not organized in sarcomeres. Also, the cells here communicate via gap junction, just like the cardiac cells, okay? And I don't worry too much about this one, okay? Now, when we talk about contraction of smooth muscle, a smooth muscle that receives innervation from, of course, the nerve tissue, at the level of an axon terminal branches out just like it happened for the skeletal muscle, but instead of the typical synapse that we have seen that forms, there are these enlargements, basically these enlargements of the branching of the branches. And these are the points at which neurotransmitter may be released, okay? Same type of neurotransmitter that we have seen for the skeletal muscle, but also other types, okay? So here I want to appreciate that the myofibrils here are present, and the same type of myofibrils, including myosin and actin fibers, actin sliding over the myosin, same thing. They're just not arranged so regularly that you can appreciate striation, but they are arranged more diagonally. And between sets of myofibrils here, you have these points of connection. Remember, they have to be all connected because they have to communicate and contract together at the same time. There are these structures here known as dense bodies. Okay, they're made of proteins as well. All right, so now, what elicits contraction of smooth muscle tissue, smooth muscle cells, is the same exact thing that we talked about for the other two types of muscle tissue, is calcium. But the way calcium is released is different. Most of the calcium, in fact, is released at these caveoli, which are indentations of invaginations of the plasma membrane present all throughout the sarcolemma. They don't go down into forming T tubules, There are no T-tubules here, but there are all these invagination. But if you will, this invagination correspond to the T-tubules of the skeletal muscle. And here there are calcium channels, and most of the calcium that releases contraction here comes from the outside and of the cell through these channels that are present in the caveoli. Okay, so it comes from the extracellular fluid. This is a fundamental difference with the skeletal muscle, because remember that the calcium in the skeletal muscle fibers is released from the sarcoplasmic reticulum. So it's already inside the cell, although it's kept away in a compartment, but it's already inside. Here, instead, it comes from the outside, from the extracellular fluid. And the mechanism of contraction ultimately is the same that we have already seen. There are just a little bit few steps upstream, if you will, where what happens is that when the calcium is released, it comes into the intracellular fluid. It binds to a specific protein that is in the cytoplasm, present always in the cytoplasm, is known as calmodulin. And there is a complex that forms, so calcium calmodulin activates the myosin kinase, which is an enzyme that phosphorylates myosin heads. Phosphorylates them. Remember that also the myosin heads in the skeletal muscles were also phosphorylated. This phosphorylation event, it liberates energy, and now myosin can move and bind with actin. The myosin heads can cock, right? And so this is what I just told you, okay? Calcium comes mostly from the extracellular fluid. A little bit also comes from the sarcoplasmic reticulum, I have to say. And this happens in response to, again, a neurotransmitter release from nervous system, but as we will see, there are other signals as well that can elicit smooth muscle contraction. I have it in one of the next slides. Those are very important to know. Now let's review again the smooth muscle contraction pathway. So calcium binds to calmodulin, forms this complex. This complex activates the kinase, okay? And you can just call it myosin kinase, okay? I know here it says myosin light chain kinase. And therefore, phosphorylation of the heads of the myosin make it move to where it engages with the active sites of actin, okay? And here it's not shown, but there are proteins that are also binding to actin and masking normally the actin active site. They're not the same proteins that we talked about for the skeletal muscle tissue. They're different proteins, but the mechanism is the exact same, and they will get out of the way basically in order for this contraction to happen. I don't think I have it here. Let me see. Yeah, I don't have it here. Okay, don't worry about it, but you know, it's the same type of mechanism that we saw for skeletal muscle, but not the same, not troponin and tropomyosin, other types of proteins. All right, so here is a comparison, okay, between the two mechanisms of contraction between smooth and skeletal muscle. Now, just briefly, I want to talk about the physiology of smooth muscle tissue contraction. Smooth muscle doesn't contract as rapidly as skeletal muscle, so the contraction starts slower and lasts longer. Now, this is important because typically, of course, there could be a few exceptions, but and this is important, especially for that kind of smooth muscle that that surrounds the gastrointestinal tract or the blood vessels. Remember, those are organs that must maintain a certain tone, a certain pressure there, and therefore you want that there is a constant prolonged contraction of those types of muscle fibers. Okay, and the reason why that a type of a contraction is slower and lasts much longer is due to the particular type of calcium release into the cell, and also the fact that the calcium stays in the cell for much prolonged time. It moves out of the muscle fiber more slowly and therefore is able to activate the contraction for longer time. Here are the other important regulatory factors for contraction of smooth muscle, not just action potential from the autonomic nervous system, but also stretching, other types of hormones that are released in the blood, changes in oxygen level, carbon dioxide, pH, and temperature. Okay, this can all stimulate contraction in the smooth muscle tissue. So very important to know, okay? And I was telling you that one of the neurotransmitter involved in smooth muscle contraction is in fact acetylcholine, but other types are, for instance, epinephrine or epinephrine, they also can play a role. Here is a summary, okay, of your 3 types of muscle tissue that we talked about. Visualize this, please, for me, okay? I've told you many times, okay? And then we looked at the anatomy of skeletal muscle. We tried to understand how we define arrangement of muscle fascicles, okay? They're pretty straightforward. They're all described here. And then we described insertion, origin, and these are important. You have to know what is an insertion, what is an origin. And we described, of course, tendons and aponeurosis. The difference between the two is pretty obvious. Okay, this is just kind of rope-like structure. This is a more plate-like structure. All right, to be known. And we described agonists and antagonists. I may give you a picture like this to tell me which one is the agonist, which one is the antagonist. Very easy. Agonist, the one that is contracting. The antagonist, the one that is relaxing. Okay, in a pair. Then we talked about fixators and synergist muscles, also the definition to be known. And then we before looking at names of muscles and try to name them and recognize them, we understood that there are general lines that we can go by to kind of try to help us memorize those names, okay? And then we looked at many different types of muscle which are important. Some of these may be in the test. I'm gonna go very quick here because this is just for you to visualize. Okay, so all right, so very important muscles that could be in the test. All right, we're done with a review of our muscular system. We are spending much more time than I would like to, but hopefully all what I'm doing will help. All right, so now the first lecture of the nervous system was really an introduction. We talked about the functions, the anatomical divisions. We looked at nervous tissue and all the types of cells. We looked at synapse real quick, and we talked about reflex arcs. This is all important material. All right, the functions, okay, of course, is always one of the most important thing to know, okay? Then we talked about the anatomical divisions. We talked about the central nervous system, okay, and the peripheral nervous system. Central nervous system includes brain and spinal cord. We just talked about all this last time. And where does the spinal cord begins, okay? Remember where it begins. This is important, okay? And and then we looked at the peripheral nervous system, okay? And this consists of all the nerves, basically the nerve axons outside the protection of the skull and the vertebral column. We haven't talked about specifically about peripheral nervous system just yet. That's something we're going to talk about starting next week. All right, we talked about the pairs of nerves that come out from the cranium as well as the spinal cord. Again, we're going to talk about this more in detail. We're going to pretty much learn about all these names starting next week. Then we talked about, so these were the anatomical divisions, and we talked about functional divisions of the nervous system. We talked about the fact that there are three functional categories that we can recognize. There is a sensory, an integrative, and then motor, okay? And of course, let's look at those in detail. You have to know all of these, okay? Sensory functions gather information from the external and internal environment of the body. The input is gathered by an afferent division of peripheral nervous system. So there is an afferent or sensory. Remember, these are synonyms. We always use them interchangeably. Okay, and this is divided in somatic and visceral division because it's subdivided. Because of course you can have you have two roots here. One that is comes from your skin or skeletal system, the other that comes from your visceral organs, okay? And so there is a somatic sensory division and as well as a visceral sensory division, okay? In the sensory, you can say in the sensory division of the nervous system. All right, so then we have the integrative function. We're talking about functions here, okay? We're classifying nervous system parts based on function. Integrative functions analyze and interpret incoming sensory information, okay? And of course, these are located in the central nervous system. We talked about these integrative divisions and areas in the brain last time. Remember, I'm going to go over them again today. Then we have the motor functions, and here again we have to make a subdivision, just like we did for the somatic sensory division. For the sensory or afferent division. Okay, we're talking about motor division, is also known as efferent. Okay, these are synonyms. As we did for the sensory division, we have to subdivide in somatic and visceral motor division. Okay, and here we can recognize really two different systems, if you will. One is the somatic, of course, voluntary, and the other one is the autonomic. Okay. And of course, the autonomic is the one that regulates those involuntary, the movement of those involuntary organs, and especially, of course, we're talking here about smooth muscle tissue or cardiac muscle tissue, right? All that muscle tissue that we don't regulate the movement of, okay? So cardiac and smooth muscle tissue are the ones that we refer when we talk about the visceral motor division. Okay, regulates the movement of that kind of muscle. All right, when we talk about autonomic motor system, we can then also recognize two major subdivisions there. We talked about the sympathetic and the parasympathetic. We like to make this distinction because the two systems here are very different. One prepares for fight and flight, and the other for rest and digest responses, okay? And these are pretty much opposite responses, okay? They cause, basically, they act upon the same organs, but they cause pretty much opposite effects, okay? And for instance, in a fight or flight type of response, your heart rate increases, whereas in a rest and digest, it decreases. And rest and digest, it tells you that there is a stimulation of the intestinal activity, whereas here, where it is in a situation of emergency where you have to probably run away, then you want to pretty much inhibit activity of intestine. You don't want to take energy there, okay? So these are to be known. And this was a summary, so visualize all this. This is all we have talked about. Then we talked the nervous tissue. We described neurons, okay? Neurons have a body and processes. The short and branched one are called dendrites, and the one very long that sometimes can have collateral is the axon. We described also a few features such as the axon terminals and the telodendria, and then the axon hillock. This region is the region where action potential is initiated. And at the level of axon terminals, we can recognize synaptic bulbs, okay? This axon terminal forms a little bulb. We then describe the three fundamental regions in a neuron, which is the receptive region is the region that contains the dendrites, then there is the conductive region, and then there is the secretory region, because that's the region from which the neurotransmitter is released. We describe the three types of neurons. Remember that 99% of all your neurons are multipolar, okay? But there are also other types of neurons, the bipolar and the pseudo-unipolar. You have to be able to recognize these three for me. I may give you pictures, okay? And remember which one is the most common. It's the one that we just described basically in the previous slide. And all right, so and when we talk about these three types of neurons, these are see the types of neurons that will either be sensory neurons here. These are typically either pseudo-unipolar or bipolar. You see that this is what we looked at in the previous slide. Then there are the interneurons, which are typically multipolar. See, this is the typical neuron. Okay, this is a multipolar, as well as the motor neuron is multipolar. Okay. So where do we find the various types of neuron? Where are they function? Okay, the sensory are usually pseudo-unipolar and bipolar. The others are multipolar. We talked about synapse. Okay, synapse is the establishment of a communication. It's not a fusion of two cell membranes. There is always a gap. Remember, as much as we appreciated it in the neuromuscular junction, here the point of connection between two communication, we should say, between two neurons is the synapse, and we can recognize a presynaptic neuron and a postsynaptic neuron. Information goes this way. Okay, and so the axon of the presynaptic neuron takes it communicates with the dendrites of the postsynaptic neuron. Pay attention to this, okay, to what I just said. The axon of the presynaptic neuron communicates with the dendrites of the postsynaptic neuron. Just visualize that in your mind, make a sketch, okay, because it's very important and fundamental for you to know that it never happens that two axons communicate directly with one another, okay? It's axon to dendrite, okay, and then the signal goes down. This is gonna be your axon, right? Again, what is this gonna be communicating with? With the dendrites of the next neuron, okay? So this is very important, all right? We talked about electrical synapses, which are basically gap junctions, okay? And we talked about chemical synapses. We talked about this in an introduction, and we talked about them more in detail in our other lecture, okay? In our second lecture about action potential. So I'm going to skip very quickly on this one and explain it more in detail in next lecture that I'm going to open. All right, these are the steps involved in a chemical synapse. And again, we're going to talk about in the next lecture. Then we talked about the fact that we don't just find neurons in the nervous tissue. We find many other types of cells all together form the neuroglia or neuroglial cells. There are several types. This is very descriptive, so I'm going to pretty much let you work on this. If anything, you have to remember for sure the oligodendrocytes and the Schwann cells. You cannot skip from knowing that because these are the cells that form the myelin sheaths, okay? In the central nervous system and peripheral nervous system. You must know this very well because if you don't know about the myelin sheath, you can't even appreciate what we talked about, the action potential propagation here. Why are these myelin sheaths there? Okay, so remember the oligodendrocytes are different than have the same function as the Schwann cells, but they're different. They have the cell bodies sticking out, and one oligodendrocytes can form different sheaths on an axon. Whereas all the Schwann cells, they are different because one Schwann cell, shown here, is going to wrap around just one section of an axon and form one. She doesn't have the body sticking out, so forms one sheet at one point. All right, but the function is the same. Okay, and they whenever there is this myelin sheath, this constitute the white matter that we see in both the brain, the cerebrum, and as well as the spinal cord, right? Then we talked about the other types of cells, which are basically all supportive cells that you also have to know, okay? Especially the astrocytes are super important because they form the so-called blood-brain barrier, okay? Associate this and this. This is going to be in the test, okay? So you must know astrocytes, you must know what the blood-brain barrier is. Okay, so this is kind of like they're filters. You see how they take contact with blood vessels and neurons, and they kind of they select the substances that need to be passed. Is a molecule of glucose going to be passed? Yes. Is a certain type of drug not going to be passed? Probably, okay, because there is a barrier. So not all drugs, especially artificial drugs, are going to be passed. They're going to be filtered and allowed to go into a nerve cell. There is this astrocyte, there is a sentinel cell that is going to check it out, okay, test it and see if it's not a good molecule, is not going to be transmitted to the neurons. All right, so I'm gonna skip on this. All the other cells, just very straightforward. Okay, what you have to learn what their function is. Okay, the myelin sheath. I told you many times the function of the myelination is to speed the conduction of action potential. About 20 times faster than if it was not present. Okay. Gray matter and white matter and gray matter. Remember, we talked about this. Basically, wherever there is myelination, the myelination, because it's all fat, it will give a white appearance to the tissue. Okay, and then last thing we talked about are reflex arcs. These are very important. They're programmed automatic responses to stimuli. We all have experienced this, right? And in a reflex arc, what happens is that there is a sensory division that detects the stimulus, and this goes through the central nervous system, which integrates the stimulus and immediately produces a response through the motor efferent division. Now, the central nervous system that integrates the stimulus here is the spinal cord. It does not, it's only the spinal cord or the brainstem. It does not have time to travel to the brain. Okay, so these are the components of a reflex arc. So, of course, there's going to be a receptor on your skin or skeletal muscle, and there is therefore an afferent impulse that goes through an interneuron in the spinal cord and immediately determines a response, which is pretty much involuntary. I say it's involuntary because you don't think of that movement; it just happens, but it's part of the voluntary system. Okay. So there was a video that I played, okay, about the reflex arc. This is very important, okay? And there are different types of reflexes. Some are monosynaptic with one synapse there. Some are polysynaptic, which involve multiple synapses. This is a monosynaptic one. See, only one synapse. Here there are two synapses involved. I mean, this is just, honestly, just a detail. Don't worry too much about it, okay? But one thing that you have to definitely remember is that there are the reflexes, reflex arcs can be somatic or can be autonomic. The ones that we have looked mostly at are the somatic ones, but these can be also autonomic, okay? Where here is what is activated is a visceral effector. For instance, the goosebumps that you get, those could be because there is smooth muscle or cardiac muscle involved there. Okay. All right. Now we're going to open our chapter on neurophysiology. This is a super important chapter because we talked about action potentials here. The whole story here is around action potential, okay? In order to understand action potential, you have to review the ion channels, you have to review the concept of resting membrane potential, which I just pretty much went over. So I'm not going to repeat all this, okay? You have to recall the electrochemical gradient, what a gradient is, what an electrical gradient is, what a chemical gradient is, and combine together. And I'm going to just go directly to explain gradient potentials and action potentials. And then I'm going to review the synapse, the chemical synapse, and through the second messenger pathway. All right, so this was just an introduction about the various classes of gated ion channels, pretty straightforward, chemically gated, voltage gated. These are the same ones that we have seen also in the neuromuscular junction. Then there are also the mechanically gated here. Resting membrane potential, I already told you, but just in part one of this review. And understand the electrochemical gradient, which is combination of chemical and electrical gradient. We talk about here ions. Remember that the resting membrane potential is mainly maintained by sodium-potassium pump, which is the classic pump that we have studied basically at the very beginning, okay? As well as leak channels, as we call them. They're also leak channels. But remember, this one is involved no matter what, okay, in maintaining the resting membrane potential. This is true for every kind of cell. All right, we can measure changes in the resting membrane potential with devices, changes versus becoming where the membrane potential becomes less negative is known as depolarization. If it becomes more negative, it's called hyperpolarization. Is that right? This is important. Let me give you a graph here to recognize. And we talked about graded potentials and action potentials. They're pretty different, okay? Graded potentials are local changes in membrane potential. They vary in magnitude. They can be less or more intense. And so meaning that this value here, okay, could change, could be lower, could be higher. That is not true for action potential. Remember, action potential, as we'll see in a minute, are all on events. These potentials are mediated by ligand-gated ion channels. Is that also true for action potentials? No. Action potentials are mediated just by voltage-gated ion channels, okay? So all these differences are super important and are going to be questions. Also, graded potentials are short distance signals. This is all different from action potentials, which again are mediated by voltage-gated ions, are all or none. And action potential is always the same value. The depolarization always goes to plus 30 millivolt, okay? Whereas for a graded potential can be variable. And action potentials are the same intensity in the place where they start, which is the axon hillock, and the place where they end, which are the axon, the axon terminals. Same magnitude all throughout, okay? And very rapid, very intense change in membrane potential. The membrane potential is completely reversed. The other difference is that in a graded potential, you never go in a positive, in an inversion of the membrane potential. You may go from minus 70 to minus 55. That's, that's it, kind of. You never go to a plus. Here in action potential, you go to a plus 30. That's very strong. Okay, in order to understand action potential, we have to understand what channels are involved, and there are two types of channels involved here. There are the sodium channels and the potassium channels. Potassium channels can be closed or can be open, very straightforward, whereas the sodium channels have a third state, which is the inactivated state. This is a state in which no matter what happens, sodium basically cannot pass. Okay, it's off limit. Whereas when they are in a closed state, if they happen to be open, sodium can pass through. Basically, that's the difference. And in order to elicit an action potential, you must reach a depolarization, a level of depolarization of minus 55. If that is reached, the action potential is elicited. This is known as threshold, okay? And what causes that initial depolarization is a graded potential, okay? Now, during the phases of this curve of an action potential, we recognize a phase of depolarization, right, where the membrane potential goes up to plus 30. Then it goes down. This is known when it starts going down from the peak is repolarization. Then we recognize a hyperpolarization phase, which is a phase in which the membrane potential goes more negative than the resting potential, and then it goes back to resting potential. And we need to understand what happens in all these phases, okay? And what types of channels are involved in the depolarization. In the depolarization phase, the main channels that plays a role are the sodium channels. Okay, sodium channels open in that stage. They open because there has been a voltage change caused by degraded potential, and that is the signal that opens them. And an influx of sodium starts happening. That's your depolarization. In the meantime, potassium channels start opening, and the sodium channels go into the inactive state. Remember, there's a second gate there that closes them up. Potassium starts going out, and the membrane potential starts going back towards its resting. Okay, that's your repolarization phase. And then during the hyperpolarization state, what happens is that your potassium channels keep being open, as simple as that. They stay open for longer, let's say, that they should be, and that's why you have, as you have an efflux of potassium, your membrane potential becomes even more negative than minus 70. Okay, it may go to minus 80. Remember, minus 70 millivolt is the resting potential of your neurons. You must know this value for me. That's the one that I'm asking you to remember. I'm not asking you to remember the other values for the other types of cells that we have seen, but this one is important. And you also have to know what is the value. Let me go to the next one. What is this? Okay, the peak of an action potential is always plus 13, so it does never change. Okay, so plus 30 millivolt. Minus 70 to plus 30. That's your action potential. You must know these values. All right, and here just if you want to really summarize what happens at the level of ion channels, this is your best summary graph. It shows you the various stages of ion channels, which ones are open, which ones are closed. Okay, this is the summary of what we just said. Now I want you to know very well this inactive state. When it happens for the sodium channels, this is important because this determines an important outcome and feature of action potentials. Basically, the question here is that at this level, when you're having repolarization, can you fire over here another action potential? The answer is no, right? Why that answer is no? Because these channels here would be important to open in order to fire a new action potential. But because they are inactive, you can't. Okay, so that's the key thing to remember. All right, for me, that's the very particular feature of these channels. Okay, that impedes that two action potentials overlap. Okay, so you can fire it again here in this phase, because you see that now going back to hyperpolarization, your sodium channels have changed their state. Look at the difference. Okay, now they are closed. They're simply closed. Okay, so you could go ahead and elicit another action potential here, but you cannot do it here. Why am I spending all the time here, all this time? You know why? There could be a question here in the test where you have to tell me what's happening here in terms of status of sodium channel. Okay? That is known as refractory period, what we just said, okay? So there is a period of time where you cannot, no matter what, you cannot elicit. No matter how intense of a stimulus you give to a cell, it cannot generate another action potential. And it's all this period over here, okay, of course. And past that period of time, you could generate another action potential. This is known as a relative refractory period, because even if you, yes, you can potentially give another stimulus, and sorry, this should be down here. Sorry, action potential is always plus 30. Even if you can, in reality, you would have to give a much stronger stimulus. So the stimulus cannot be a minus 55, a threshold. It has to be higher in order for a second action potential to happen here in this phase. Okay, that's your relative refractory period. All right, we talked about the factors that can speed up, not change the intensity, right? But it can actually speed action potential, so make it quicker, okay? And these are the axon diameter and myelin sheath presence. And remember the saltatory conduction, okay, of an action potential in the myelinated axon, okay? And the nodes of Ranvier's, which are the naked areas. These are the areas where the action potential is actually propagating, because it cannot propagate through the myelin sheath, okay? Saltatory propagation through the node of Ranvier's, from node of Ranvier to node of Ranvier, makes it much faster, about 20 times faster than unmyelinated axon. All right, in the second part here, we talked about synapses. We talked about electrical ones, which are basically made by gap junctions, okay? And we focused on the chemical synapses, which are the more complex one. Here is easy, it's just a channel, right? It's very, very fast communication and propagation of the stimulus. In chemical synapse, we have to recognize several steps, okay? And these are the same, pretty much the same steps that we recognize in the chemical synapse of neuromuscular junction. First of all, the action potential will actually determine a calcium influx from the outside to inside of the cell, and this causes the release of a neurotransmitter normally contained in vesicles in the synaptic cleft. The neurotransmitter binds to specific ion channels, which are the typical ligand-gated channels, and therefore the channels open. An influx of sodium typically happens towards the inside of cell. You have a local depolarization here. This is your graded potential, right? Which is mediated by these types of ligand-gated channels. And that is the trigger that triggers an action potential. All right? And remember that, just like I said, for the neuromuscular junction, the neurotransmitter needs to be eliminated, degraded pretty quickly from that synaptic cleft in order to for the situation to be reestablished to where another stimulus can fire. Now, when we talk about postsynaptic potential, there are two types. We always talk about excitatory, right? Excitation, but in reality, not always you have excitatory postsynaptic potentials. You could also have inhibition, okay? And this happens, okay? There are neurotransmitters that instead of being excitatory, they're inhibitory. What does that mean? These two are typically presented with acronyms, okay? Inhibitory postsynaptic potential, IPSP. And it's very intuitive. If I ask you what is happening at the level of membrane potential here, Of course, when you have an EPSP, excitatory postsynaptic potential, you are actually depolarizing your membrane. Is that right? It's what we talked about so far. This is caused by a sodium influx, whereas some neurotransmitters actually will instead of playing a role on sodium channels, they will play a role on potassium channels, and they will actually create an efflux of potassium from the inside the cell towards extracellular fluid, causing a hyperpolarization. Makes a lot of sense, right? You want to inhibit. There is a neurotransmitter, a specific neurotransmitter in your brain, which is the GABA. This is a neurotransmitter that basically calms you down. It inhibits synapses, okay? Whereas the most important one, excitatory one in the brain, is glutamate. At least can you remember these two for me, okay? Glutamate and GABA, but we did talk about several others. I have a table. That's interesting stuff, right? And this is what I was trying to tell you, okay? Probably I should put this slide a little bit earlier, where no matter what kind of neurotransmitter you're talking about, this neurotransmitter needs to be removed pretty quickly from the synaptic cleft after release, and this is done mostly enzymatically. This blue thing here is an enzyme that binds to the neurotransmitter and chops it up in many pieces. All right, so these are many of the neurotransmitters found in the brain. We talked about GABA being the inhibitory one, glutamate the most important of the excitatory one. This is involved mostly in learning, memory, okay, creation of nerve contacts, but we have many others. We did talk about adrenaline and noradrenaline, especially for the fight or flight response. Many others, such as dopamine, serotonin, endorphins, these are released many times in pleasant situations or when you exercise. Of course, acetylcholine. That was the first one we talked about for the neuromuscular junction. It's also present in the nervous system as well. And again, it's also a very important one. It's kind of like similar to glutamate, okay, in terms of what regulates. GABA and glutamate are the ones the very minimalistic ones that I want you to remember, that have a very important role in the central nervous system, okay? Finally, we talked about summation of excitatory postsynaptic potential. We talked about the spatial. This is a spatial summation. I'm not going to even talk about it because it's very visual. Many synapses at one postsynaptic neuron, or you can have temporal summation, which means several firing coming from one synapses of a presynaptic neuron to a postsynaptic neuron. Simple as that, okay? No matter what, the principle here is that one graded potential from one synapse is never enough, pretty much, to elicit, to reach threshold, to minus 55, which is the threshold for an action potential. So you need this situation, either spatial summation or spatial summation or temporal summation, which was on the previous slide, in order to elicit the action potential. So remember this, one synapse is never enough. You must have several, or you must have a temporal summation, okay, in order for an action potential to be generated. Here we talked about the fact that there aren't just ion channels that are involved as neurotransmitter receptors. There are also other types that we didn't talk about very much, but there are the G protein coupled receptors, okay, and these are called G coupled because they are coupled with a G protein. And in this type of receptors, there is a cascade of activation where eventually a second messenger is activated. And the second messenger most likely is CAMP. That's all I am going to tell you, okay, to remember. And here, I don't think I have introduced, these are not in the presentation that I posted, but if you want to go ahead and watch some of these are videos about action potential. So they're, again, very informative, very visual, okay? Now, last thing we talked about regarding the nervous system is the central nervous system. This is much more just descriptive, so I'm not really spending much time here. This is really a reflection of how much time you spend on this lecture, how you do in the exam. Central nervous system is composed by brain and spinal cord. We talked about functions, of course. Gray versus white matter and its distribution in the brain versus the spinal cord is inverted. You cannot miss that, right? And what is the gray matter? What is the white matter? I already told you in the previous lecture. Gray matter, remember, is all the cell bodies, the neuroglia, and unmyelinated axons. White matter is pretty much myelinated axons. We talked about the several divisions of the brain: cerebrum, the encephalon, cerebellum, and brainstem. You kind of have to know all the portions of the cerebrum, from the cortex, corpus callosum, basal nuclei, limbic system. We talked about all this. Of course, you have to know about the fissures that we can recognize that divide the brain in left and right hemisphere, and the lateralization of the brain. Is the brain anatomically identical, left and right? Yes. Is that functionally identical? No. There is a lateralization, if you remember. Anyways, definition of fissures, definition of sulci and gyri, okay? And the two fissures are the longitudinal and the transverse, to be recognized. All the various lobes, which are pretty much defined by sulci. This is your central sulcus here, okay? This is another one. There are three major sulci, okay? I have them in another slide, which define the lobes: frontal, parietal, occipital, temporal, and then there is the insula. Pretty straightforward. They take name from the bones. And remember, there is a lot of vasculature here, and then we learn about the names of the three sulci. Remember, sulci are more shallow than fissures. There is the central, there is the parietal, occipital, and the lateral. Okay, central, parietal, occipital. Lateral, and occipital or parietal occipital. And in a section of the brain, in a frontal section, you can appreciate, and the right section actually, the basal nuclei, which are spots of gray matter inside the white matter. Pretty obvious. We talked about the central cortex. We talked about all the specific areas or the domains, sensory, association, and motor areas. You have to know these two areas for me very well. This major sensory area, and this is your major motor area, okay? We talked extensively about this is the primary somatosensory cortex, where all your sensations and sensory information from skin and skeletal muscle comes to, okay? Then you have your primary motor cortex here, which is all what directs, is a gyrus, right? That directs all your voluntary motor movements. And remember that for both somatosensory and motor cortex, we go to the homunculi. Basically, that's what your brain sees, okay? For sensory and motor. May give you these homunculi in the test to tell me what they signify, okay? And so a lot more sensory importance in terms of amount of cortex is dedicated to these areas of the body, as well as for your motor functions as well. Okay. And so we talked about the Broca's area. This is also important. It's only present in the left portion of your brain. It's involved in speaking ability. I want you to know that one. That's an important one. You should know it as a general culture thing also. And the one thing I want to highlight is this as well. Remember that most of your sensory information travels through this portion of the body of the brain known as thalamus, which we talked in detail about at the end of this presentation, except for the smell. The smell, sensory information of smell does not go through the thalamus, okay? That's the one exception. And we talked about the association areas. There are three main association areas. The limbic one is very much involved with your emotions. And then there is the, which is over here, then there is the anterior and the posterior as well. And they have different, these three different association areas are not only physically recognizable, anatomically distant from one another, but also are involved with different functions, okay, as we saw. And we talked about lateralization, brain lateralization. You need to know the concept. And if you could remember for me the more evident, important functions of left versus right hemisphere. Remember, left is more involved with logic. Okay, remember that, where right is more involved with emotion. Okay, that's pretty much the main difference. 90% of humans have a left-sided dominance. And remember that, so there is a dominance, basically, meaning that really there are more connections there in the left side of the brain. And remember, the left side of the brain, things crisscross here. Left control the right portion of the body and vice versa. So if you have a left sided dominance, you're gonna be hand and you're gonna be writing with your right hand, okay, because you're left side dominant. People that have they're left-handed, they have it reversed, and these are much less, okay, in percentage. All right, this was just a summary, and then we talked about the corpus callosum, basal nuclear limbic system. Again, this is all descriptive. Corpus callosum is very recognizable, okay, especially in picture. And basal nuclei, and of course their function here is all white matter or axons that travel from left to right, putting connection left and right brain hemisphere. And basal nuclei, we name them. I told you, don't worry to remember all the names of the specific ones. Just remember pretty much what they are and what they are involved in. When we talked about the limbic system, which has three components to it, you have to remember these three components. Generally speaking, we can say that the limbic system is involved in production of emotions and learning and memory as well, generally speaking. Then we talked about cerebellum. Cerebellum is a very recognizable structure. Sorry. System. No, we talked about the diencephalon first. The cerebellum is very straightforward. Okay, it's down here. This one, we can see it in section. We're going to talk about the diencephalon first. And the encephalon contains the thalamus, this very important station through which all the sensory information except for the olfactory one travels, right? We saw that in one of the previous slides. The hypothalamus and the pineal gland, which is this little gland over here. And we talked about the thalamus being a relay station for all ascending sensory information, except olfaction. Okay, I've told you already 3 or 4 times. This is important. This is in connection with the hypothalamus. And so, thalamus, yes, is a relay station for all sending information, but it's also involved in motor control by reinforcing. So it also goes the other way, information. So involved in motor control by reinforcing voluntary motor behavior. And thalamus is in a connection with hypothalamus, which is in connection with the pituitary gland, which is the mother gland of all your glands. So here there is a connection between nervous system and endocrine system, okay? And all the functions of the hypothalamus, pretty much hypothalamus is pretty much who you are, okay, to be known. And the pineal gland, very simple. This little gland over here, there is one thing that it does. This is a gland. It's not part of the nervous system, but it is immersed pretty much in the nervous system and in connection with it. Secretes melatonin, which is involved in the regulation of your biological clock. Cerebellum, as I was telling you, in section is very recognizable, is involved with mostly maintaining the balance of your body. Then we talked about the brain stem and the various portions, midbrain, pons, and medulla oblongata. Medulla oblongata, remember, is the site of where all nerves come out for regulation of your heartbeat. I told you when we were talking about cardiac muscle, okay? And the various functions of the midbrain, and then the pons, which contains the reticular formation. That's the main important thing, and the medulla oblongata to be known. Okay. Then we talked about the anatomy of spinal cord. It is straightforward. Okay. And basically, we're going to talk more about the nerves when this is part of the peripheral nervous system. This is central nervous system, right? That's what we are focusing on, but they're connected. And so we can recognize here the horns and the funiculi. The horns are in the gray matter, the funicular are in the white matter. And in the dorsal and ventral horn, we can recognize the bodies, the cell bodies of different types of neurons, okay? And remember that the dorsal root contains the somatic sensory nuclei, and of course the processes, whereas so the sensory, okay, whereas the ventral root contains the motor, okay, and both are somatic. So this is the here. This is where all the sensory information from skin and skeletal system arrives. This is where all the input for movement of skeletal muscle comes from. Okay, so somatic. But in between here, this is your lateral horn. And this is another area that we find in between. Here is your autonomic efferent cell bodies that regulate, of course, those stimuli that go to smooth and cardiac tissue. Okay, and here is your visceral, here is where your cell bodies of your visceral sensory information comes to. All right, so here is explained a little better what I just said. Here more visualization. And he actually, the one that I like the best is this figure, because it summarizes what I just told you, I should probably have shown you this, okay? So no matter what, the dorsal or posterior root is pretty much a sensory root, whereas the ventral or anterior root of your nerves is the motor one, okay? And so you have the somatic and visceral respectively here in the posterior root, and then you have the starting from the more anterior one, the somatic and autonomic roots. I'm a little bit tired, so I think this is one of the last things I told you. Oh yeah, we talked about the ventricles of the brain to be known, okay? This space is filled with the cerebral spinal fluid, which regulates and maintains the stability, chemical stability of the external environment of nerve cells, but also gives protection, cushions from inside your central nervous system. And then we talk about the meninges, okay, which are these membranes that actually protect the brain and the brain vasculature. So we talked about the pia mater, arachnoid mater, and the dura mater, which is the one that is really closest to the scalp. All right, and I think that was it. And hopefully this review is going to be helpful. And I told you what are very important things to study, and they're going to be in the test, okay? I wish you very good luck on your test. Thank you.